Hi, everyone. Thank you, Joanne, very much for the nice introduction. First of all, good morning, good afternoon, good evening, everyone, whoever, where you are in the world. Maybe you guys have some nice coffee. I'm having actually my tisane. It's 9 p.m. here, so it's fine, no problem. So again, welcome, and I'm really honored to talk to you about today at this session on health-based exposure limits, HBELs in a shared facility. In this session, I will discuss the importance of having established solid HBELs in your shared facility and the challenges, of course, that you may encounter. So before we dive into the subject matter, I would like, first of all, to introduce you to Agility, for which I'm the toxicology program manager. If you see the nice image here, this is Montreal. It's dear to my heart, so that's why you see a nice picture of Montreal over there. This Agility solution was founded in 2002 by our CEO, our dear CEO, Dean Calhoun, who is a certified industrial hygienist. We have over 800 active clients in over 70 countries right now. And also what's fun with Agility is that we're located a bit everywhere in the world. I mean, we are from home, we are a home base, but we're virtually everywhere. So we're located in the United States of America. We're located in Singapore, where Joanne is right now. We're located also in United Arab Emirates, in Canada, myself. And we have our very dearly toxicologists, my toxicologist colleagues who are located in Brazil. Our primary services that we do right now, it's primarily OEL, ADE, PDE monographs. We also do occupational health categorization, which are OHCs, also known as OHCs. We'll look at it a bit further on. We also create safety data sheets for different compounds. And of course, we also have other services: genotoxicity, impurity assessment, excipient justification reports, residual solvent reports, and containment justification reports. And all our reports are certified by a certified toxicologist. So it's very important. And if you want to have a look at our site, you can always visit agility.com. It's our website where you see our catalog, which is, I think right now we have over 200 compounds already established HBELs for them. And you can see all the services that we have. So now let's dive into the subject of this session. So I would like, first of all, to look at some important definitions. I would like to look at some important definition related to HBELs. So first of all, of course, we have the occupational exposure limit, which is also known as OELs. So an OEL, I'm sorry, very sensitive. So an OEL is defined to be an airborne concentration and by the way, this is from the, if you see the source, it's a very standard definition by many regulations. And it's right now it's by the ISP. So an OEL is defined to be an airborne concentration which nearly all workers may be repeatedly exposed to for 8 hours a day, 40 hours a week, without adverse effect based on currently available information. It does not take into account hypersusceptible or otherwise unusually responsive individual or person with hypersensitivity to a substance, which may be exacerbated by exposure to this compound. So this is very important to know because sometimes, and like we all know, in shared facilities, an employee can be exposed repeatedly to the same compound, 8 hours, usually a shift, it's 8 hours a day, and it's a 40 hours a week. So very important to establish solid HBELs in order to protect the worker. Another important measurement in the HBELs, it's the acceptable daily exposure, also known as ADE, which is also known as the permitted daily exposure, also known PDE. So the acceptable daily exposure, or ADE/PDE, is defined as a dose that is unlikely to cause an adverse effect if an individual is exposed, and it's important here, by any route at or below this dose every day over a lifetime. So very important. So there's two distinct two distinct definition and two distinct measurements for the HBELs. All right, and I wanted to mention this because we often see this mistaken. So we have to understand that OEL or ADE are usually based only on the toxicology properties of the compound. We have to differentiate here between the risk and probability of exposure. Why am I asking you this? Why am I saying you this? Is that often we see questions like, oh yeah, but in our formulation, the compound is very low. I'll give you an example. In a shampoo, in a bottle of shampoo, you will find alcohol. Alcohol will be one of the many components that you will see in the back of your shampoo, bottle of shampoo. I know it's a simple example, but I just want to, more concrete example. So you have, like, for example, 2.5% in the compound. The risk for you to be exposed to alcohol is very low. But the hazard of the compound, you have to understand that it's still based only on the toxicology property of the compound. So it has nothing to do with external factor, with the formulation of your mixture. So again, the hazard of the compound is based only on the toxicology of the active pharmaceutical ingredient, and the probability of exposure is based on many factors. Here I mention you a list. It's not limited to this, but you could have your scale of process. So you have to think, am I doing a small batch or am I doing a huge batch in a shared facility, a commercial batch that you're going to do your drug, or you're doing just a little pilot batch? The physical state of your compound, is it dry solid? Is it liquid? Is it wet cake? Among others. I mean, when you work with a powder, you have a lot of volatile particles in the air. So you're more, at risk to be exposed to than in a solution where everything is already wet and it's in a solution. So you don't have that many particles around you. The amount of energy imparted on the process, the engineering control used. So do you have filters, anything that would help to contain? The frequency of exposure, the duration of exposure. The duration of exposure meaning you have a manufacturing process and you have weighing, you have mixing, you have packaging. In a weighing, you could pass 15 minutes, or on your mixing, you could pass half an hour, an hour and a half. Encapsulation, so different duration of exposure during your manufacturing process. Percentage of the API again, and the formulation, and many others. So just to make sure, you could use always the equation when you think about it, risk equal hazard multiplied by probability of exposure. It will give you the risk of exposure, not the hazardous property of the compound. Let's look also, by establishing HBELs, you also have some requirement and some regulatory agency that it's controlling, that it's evaluating. Are you having in your facility, are your HBELs well defined? Is it, are they regulated? So a lot of agency will be, for example, you'll have Health Canada, you'll have Food and Drug Administration, also known as FDA. You will have the European Medicine Agency, EMA. You also would have guidelines such as risk map established by ISPE, which helps for cross-contamination in your products. So those are regulations that are very important to respect. And honestly, more and more we see that it starts to be more regulated than it used to be. So you gotta be careful, and you gotta make sure that you have your compounds are really well evaluated and they're solid. I mean, the HBELs are solid, and they're done accordingly. Also, a question that often we see, are they necessary? Are they required? Or is just an optional thing to do? No, it's not optional. You have to have HBELs required for all medicinal products. So you have to, it's a requirement. It's not optional. And this is from the EMA, the Q&A from EMA, but you also have from Health Canada, you have from FDA. It's not only EMA that requires that, it's all of them. Another thing is in a shared facility that could be very complex, that could be more complex than in a dedicated, environment, which you have one line is dedicated to one product. So your risk of cross-contamination is less. In a shared facility, the problem is that you could have certain antibiotics, certain hormones, cytotoxic compounds, certain highly active drugs. They're very hard, and this, you have to be careful when you share this in a facility because usually it's not really accepted. And for certain antibiotics, we're talking about sensitization. An employee that could be sensitized in a facility, for example, it can happen that it cannot even approach the facility because he's already sensitized to the product. So it could have, a lot of impact, and you could also have cross-contamination in your product. So now, who's considered qualified in establishing HBELs? So again, HBELs is, we also I attended the webinar. No, you need a bit more than just attending webinars. You could attend and you can inform yourself, and you could learn about HBELs. But in order to have established HBELs, you need someone that has experience under their belt, many years in the pharmaceutical industry. For sure, it's a plus if you're certified. So because you already proved to agencies that your experience is deep enough and solid enough in order to evaluate those compounds and establishing HBELs. Because don't forget, we do this all for protecting workers and protecting the products, to have a safe product at the end of the line, which we send to a patient, right? So what it says here, and this is from the ISTM standard. So, and again, this is from the ISTM standard, but and a lot of regulation is the same thing. They ask you to have, first of all, your curriculum vitae has to be always available, demonstrating your educational background. So for example, toxicology, pharmacology, medicine, or other related disciplines. If you're coming to tell the regulator, yes, I have a background in geography. I mean, it's nice. It's a good background to have, but it's not very related to the field, So again, you really have to have something that is very related to pharmacology, medicine, or other health-related disciplines. Certifications such as, like I said, the Diplomate of American Board of Toxicology, DBT, or European Registered Toxicologist, ERT. These are important because, again, it demonstrates that your background is solid enough. It's a plus. It's not obliged anywhere to be certified, but it's a plus, and it shows your background. Also have years of experience in the field and publication related to the field. So while all are not required for a qualified expert, the appropriate documentation in this area demonstrate the expertise to work in this area. Typically, certification registries require an academic, like I told you, academic degree in a relevant subject. So again, you're showing already to the agency that you're qualified. So it's a bonus. It's a plus, It keeps you more it keeps you more in confidence that you have the knowledge to do this. And by the way, all our reports, like I told you, Agility Solution, we're all certified. So, which is very good. They're all signed by a certified toxicologist. Again, so why exactly should we care? Why should we care that, to do some HBELs? Why is it important to have those HBELs in place in a shared facility or in any facility in the pharmaceutical industry? So HBELs are critical in ensuring the safety and health of employees, of course, who work with materials that can be hazardous for their health. As they're established based on the toxicological properties of the substance being handled, it provides a level of exposure that it's considered safe for an employee and the product. In other words, if you have an active pharmaceutical ingredients that are not periodically evaluated and controlled, occupational exposure can happen, and also cross-contamination issues in your facility, which will then have findings and then recalls and then go on and on. And an example of that, it's not very a funny example, but an example of that I saw in my previous experience in pharmaceutical industry, I'll give you a compound, mirtazapine. Mirtazapine, it's an atypical antidepressant. When people were working that, and that's before we put the HBELs in place. That was before we put the contrabanding and we started to put everything in place. People, after they were working in the manufacturing facility, they were falling asleep. They're literally sleeping in the hallways after working with this. So you understand the adverse health effect that could happen. Even when you go home, you could fall asleep while going home, driving home. So it's examples like that, you know, those people who work directly with the compound, you have to be careful because it's their health. It's their health, and we care about that. And also the cross-contamination of your products. You can have cross-contamination between your products. And this, again, you don't forget, you're giving your products to patients. Patients will ingest, you know, a compound that is probably contaminated with something else. So we got to be careful. I'm really apologizing for this. I think my mouse is very sensitive, so it's go back and forth. Sorry about that. Okay, so when we talk about establishing HBELs, you have compounds that are challenging in terms of occupational exposure, and we'll look at the next slide, which are compounds that are challenging in terms of cleaning capabilities. So cleaning the equipment, cleaning the surface after you work with the compound. Example of compounds that are very challenging in occupational exposure that we saw is compounds that are known to cause cancer, such as benzene, formaldehyde. You also have all those alkylating agents. Those are compounds such as cyclophosphamide and cystoplatin, which are known for cancer therapy, so chemotherapy. So those are very particular challenging in terms of occupational exposure. Exposure to these compounds can increase the risk of developing cancer, and therefore strict control must be put in place to limit exposure. You also have compounds that could change your DNA. So it could change your DNA leading to genetic mutation. That could be challenging also. Compounds such as, you know, nitrosamines. Nitrosamines right now, it's a hot topic. These compounds, you know, are example of mutagen that could be very difficult to control in the workplace. Teratogens are compounds that could cause birth defect. We all saw the case with thalidomide, which you would have deformities in children, and moms that would take that compound, you'll have very dangerous birth defects. Also some compounds such as colchicine. Colchicine are highly toxic and can cause severe health effect at low level exposure. I took this example because I think this compound is one of the compounds that the dose is so low to cause death. It's 7 milligrams, the toxic dose. So it's a very small dose. So they're very, very hard to work with. You have to have strict control in place and protective measures in order to prevent exposure to an employee. Compounds are challenging, like I told you, in terms, you also have cleaning. Cleaning, it's important for cross-contamination. You want to make sure that you clean all your traces of the product that preceded the other, right? You don't want to have trace of one product to the other and then it goes to the patient. So compounds that are hard to clean, such as lipids and oil, can be challenging to remove due to their hydrophobic nature. Specialized cleaning procedure and detergent are required to effectively remove lipid and oil residues from equipment. Residual solvents, such as those used in the manufacturing of APIs, active pharmaceutical ingredients, can be challenging to remove during the cleaning process. They're volatile. They have a low solubility in water, making them difficult to detect and to remove. You also have highly potent compounds which are considered worst case compounds for cleaning validation due to their low ADEs. They become their worst case and their potency. Even small amounts of these compounds can cause harm, make it crucial to ensure that all traces are effectively removed during the cleaning process. This is very important. Cleaning is also a it's a very challenging part of the manufacturer industry, but it's also very important. Again, the expression that we all know, bad habits die hard. They do even in establishing HBELs. So what I'm saying about that, why am I saying about that, is that animal studies are often used to establish HBELs, but they have limitation in predicting human response to exposure. Relying only on animal studies can result in an accurate assessment for the risk to human health. I give you an example. You have animal studies, because don't forget, why are we doing those HBELs? To protect your work and to protect the product, right? So you have to make sure that when you do your calculation, you have to take this in consideration. You cannot always base yourself on animal studies when you also have clinical. We're doing this for humans. So if you have a more appropriate information regarding dosing and clinical information, then you could go with your clinical information instead of going only with animal studies. Not all individuals have the same susceptibility, to hazardous substances. Some people may be more susceptible due to genetic factors, age, or pre-existing health condition. Ignoring these differences can result in an underestimating of the risk to human health. So when you do your HBELs, you have to take this in consideration, that there are special cases and that not everyone reacts to the compound the same way, So you have to take this in consideration. Also, the establishment of HBEL require up-to-date and comprehensive data of the substance in question. Using outdated or incomplete data can result in inaccurate assessment of the risk to human health. That means, compounds, there's some old compounds that are marketed for a very long time on the market. And they're studied for other new indications. New literature came up, a new indication, it was approved for something else at different dosage, new animal studies, new assessments, safety assessment were up there. So you always have to make sure that you're up to date. I call it literature monitoring. So you have to make sure that, you're up to date with your, if you have an HBEL that was established in 2014, you have to think about it, that you need to reevaluate your compound. It's about time, because literature always evolves and you're gonna have new indication on the market. So yeah, better be careful and make sure. And again, you have to have the knowledge for this. You have to have the knowledge to know what you're looking for, what kind of study, what are the reliable sources. So that's why they're saying, in order to be qualified to write that, you have to be experienced. Even if you're not certified, you still have to be experienced. Attending a webinar, it's very nice. You inform yourself, but not enough. Not enough to establish HBELs. Also, the establishment of HBELs should consider all possible exposure of routes, including inhalation, ingestion, and dermal contact. Failing to consider all exposure routes can result in an underestimated of the risk to human health. And again, believing numbers, and this I see it all the time, believing numbers without understanding their basis. Here it is. You have a nice, I give you the OEL and the ADE for different compounds. So you have citalopram and entecavir, which is a very well-known product, minocycline, paliperidone, and acarbose. And I know right now, a lot of you maybe are taking picture of the slides or taking notes of the OEL and ADEs. But guys, all numbers on previous slides are fictitious. They're completely fictitious. They're not real. It's just to show you that believing a number without having the assessment behind it, it doesn't help you. It really doesn't help you. I see often, I just want to have the OEL, I just want to have the AD. Can you just give me this information? No, because you need to know what's the what's the knowledge behind it? What did you do in order to come to this number or to come to this HBELs? Because it's important. I mean, you're going to go to regulators and you say, oh, I have my OEL, here it is. The regulator will ask you, where's your assessment? An OEL, you know, everybody could write an OEL, put the number on the paper. Totally, you have to make sure. So again, I know you guys probably maybe some took pictures, but because we're all humans and we all have the same, like we say, bad habits never die. So our numbers on previous slides are fictitious. So when we say establishing HBELs, it doesn't come without challenges. Challenges often encountered in established HBELs, or for example, I'll give you some compounds, investigational compounds. Investigational compounds are usually novel compounds, preclinical phase that were never tested in humans. They were never tested in humans, so the data is very limited. You have a very limited information, toxicological information, in order to establish an HBEL. It's difficult. This can be particularly challenging, like I said, for new or emerging substances, such as, for example, now we have a lot, it's a hot topic, vaccines, monoclonal antibodies. The information is still very limited. What we usually do for those types of compounds are those occupational health categorization reports, the OHCs. It will give you a range for your OEL, and the ADU will based on the OEL, but it's very challenging because you don't have much to play with. So you have to make sure, you know, you have some critical parameters that you can evaluate in order to establish an HBEL. Another one is mixture of pharmaceuticals, powders, mixtures. The toxicity of mixtures can be complex and depends on the interaction between the individual components. So this toxicity of mixtures, you can have some ingredients that are more potent than the others. You can have mixture in the mixture, some compounds in the mixture, you don't know what they are. You don't have the information. And you have to know which component in the mixture is more potent. How would you evaluate your HBEL based on which component of the mixture? So it could be at the end very complex in order to establish HBEL for the mix as a whole. And again, a class that it's very challenging right now, and I like it. I like to evaluate those because you have very interesting stuff out there. So challenging often encountered in natural health products, so also known as NHPs. First of all, inconsistent application of the definition of NHPs. Different regulators have different definition. What is an NHP? What is a not qualified as an NHP? So it could be very challenging actually to understand, is it an NHP? Is it not? Changing people mindset that natural equals safe. And we will see case studies after the slides of compounds that are not that as safe as we think they are. Determining the most critical, like I said, critical ingredient in the NHP. In a plant, you can have 20 components. EMA assessment may be only available for a minor component. Safety information is often not available. Sometimes they're not approved. They're claims. So it's claimed to benefit you for this, but it's not an approved drug. It doesn't have like the new drug application package. It doesn't have a package insert. Often you don't have SDSs available, safety data sheet for those. So it could be very challenging. So let's look at some cases. Interesting compounds. So one of it is Icelandic moss. So this compound, it's advertised for medical uses such as dry cough, upper throat irritation, inflammation of the upper respiratory tract, many others. So it comes as gummies, as lozenge, as syrup, even as herbal tea. You can drink this. An EMA assessment was completed in 2014, and we're gonna explain you why in the next slide we decline actually to establish an HBEL for this type of compound. At Agility, we decline to do it. We are very curious at Agility Solution. We're a team that is very curious. If you want to know more about, you could look, I have here it's a nice assessment of toxicity on Iceland lichen, which is the Icelandic moss. You can look at this article, it's very interesting. It gives you all the toxicity information on the compound. And as we are very curious, what we did, we went online, we bought 4 different brands of this. 4 different brands, and we analyzed them in an independent laboratory. What we have found out was kind of alarming, yes, because all 4 products contain lead above the Centers for Disease Control and Prevention interim reference limit for lead. So in addition, all 4 products were above the California Proposition 65 maximum allowable dose level for reproductive, for example, for reproductive toxicity. It's lead, so you're gonna have a reproductive hazard. It should also be noted that none of the product label had any sort of warning. This product loves to absorb heavy metals. It has no warnings on the product label that this might contain lead when you're really over the limit by the Californian Proposition 65. And just for your information, because I'm not sure if everybody knows what California Proposition 65, it is a law in the United States that require businesses to provide warnings to Californians about significant exposure to chemicals that causes cancer, birth defect, and or reproductive harm. So again, this is why we didn't want to do it. We didn't want to do it because I don't think it's safe. It's safe to do that kind of stuff. And who tells you that when this material is being packaged on a shared facility and a shared equipment, are they testing for lead contamination? Will you have problem with your product for cross-contamination? We don't know. It wasn't advertised. It wasn't saying as a warning on the label that it could contain lead, at least, So yeah, you have to be very careful. Like I said, again, natural doesn't mean safe. Natural doesn't mean, it could be very safe, but again, not always like we see in this case. Another case study, which is very interesting, it's the St. John's wort. So this one is advertised. So you see a nice flower, it looks so safe, a nice yellow flower, it doesn't look too dangerous, right, at the first view. So the advertised medical uses, it's advertised for mild to moderate depression, anxiety, and many other claims. Advertised, not approved, but claims. It comes in pharmaceutical forms such as solid dosage form. And again, herbal tea, you can drink this. EMA assessment was completed in December 2009. And the major component in this, it's hypericin. Hypericin, it's found in this plant, and it is noted that hypericin has been shown to have toxicological properties that may cause adverse effects in some individuals. The thing is with hypericin is that it has been found to be a potent inhibitor of cytochrome P450 enzymes, which are responsible for metabolizing many drugs in the body. This can result in very dangerous interaction with certain medications, including anticoagulants, antidepressants, birth control pills. So, and it also has photosensitization. So again, natural product with an occupational exposure limit of less than 1 microgram per meter cube. So not that safe. Again, we have to break the habit to say NHP equals safe, because it's not always true. Another one that I really love, it's the famous Aloes barbadensis extract. So the aloe vera that we all know, the leaf. So it's advertised as a medical use for skin burns, acne, as a laxative, People drink this in their juice because it's very refreshing. So, it's like pharmaceutical form that you can have is solid dosage forms, cream, gels, ointment. You can have indefinite forms. There's so many forms out there of this. And again, it was completed on November, the MA assessment was completed in November 2016. And it's by the International Agency Research on Cancer. It's group 2B. So that means that it's probably carcinogen to human. Aloes that everybody likes, right? But again, if you don't use it accordingly and you expose yourself to the sun with this thing, it could have some really bad issue, and carcinogenicity was seen in mice and in animals, and knowing that it's a 2B and it's also on the Californian Proposition 65 list, it's not that safe. And it's aloes. I mean, everybody knows aloes as a good plant, that has so many benefits. It has many benefits, don't get me wrong, but you have to know how to use it. So not always safe. The other one, which is very interesting, is the apitoxin, also called as the bee venom. So, natural products from bees, usually very nice, very good for you. We hear all kinds of stuff that bees, and it's true, a lot of compounds are very good for you from the bees, but a lot of it, not so. So for this one, it advertised the medical usage, joint mobility, facial wrinkles. I like that one. Desensitization, chronic pain, and others. You can have it in capsules and you can have it in cream. Even the EMA came out with an assessment on this. But because you can have very high sensitization to this thing, the HBELs that was established by Affinity was less than 1 microgram again. So what do you say? Safe? Natural products, always safe? Not so. So again, in summary, for natural health products, we have to break the fact that safe or low risk equal natural. If you co-process NHPs in the same facility or equipment as traditional pharmaceutical, then HBELs should be established for NHPs as well. Natural health products, NHPs, can be contaminated with impurities, not normally tested in traditional current good manufacturing process. HBELs for monograph for NHPs can be difficult and lengthy, since they are usually no credible SDSs, package insert, or like for regulated drug, for approved drug, new drug application, like an NDA. So to rely upon. So when you do NHPs, here, it's very important that you know what you're doing and what sources you're looking at. Are they reliable? Because if you go to a site, advertising those nice effects, but the information, you don't know where it comes from, you don't know, are they study real or they're not. So you really have to make sure, and it's hard for NHPs, honestly, I can give you that, it's very hard. It's hard, and it could be a long process to do. Those monographs, usually, they're not 10 pages, never. So the next one, so as a summary, usually when possible, a high quality HBEL monographs should contain your chemical and physical characteristic of the substance you evaluate. You should have your therapeutic indication. You should have the clinical studies in human. It's very important. Your mechanism of action. Your mechanism of action could give you a lot of indication on the reprotoxicity or carcinogenicity. It could give you a lot of information just by looking at the mechanism of action. Pharmacokinetic and pharmacodynamics. It's important because pharmacokinetic are very important. Some compounds have a short half-life in the body. Some have a very long elimination half-life. What that means is that your compound stays in your plasma many times. So someone that is repeatedly exposed to this, it will accumulate. It will accumulate in the body you don't want that. So, and you want to be, accordingly, your HBELs have to be accordingly evaluated. You have to put this in consideration. Non-clinical toxicity studies, that's for sure. You have to have all your animal studies, whatever was done preclinical, you have to have this when possible. And also, and that's very important, your selection of point of departure. So this should be a section that should be in your monographs, because you want to see what's the logic behind establishing those HBELs. What's the logic behind your number? Why did you use that? Again, think about it. You have to use, we're doing this to protect the workers. We're not doing this just as an optional. We're doing this to protect the workers. We're doing this to protect the product by, in the end, protecting the patient who's taking this. Someone that is sick doesn't feel to have a nice contaminated product, So also you have to have a section for occupational exposure limit calculation. This is where you put all your calculation. The acceptable daily exposure calculation also you have to have included. And again, remember, acceptable daily exposure is the synonym of permitted daily exposure. It's the same thing. The regulators are speaking two different languages to say the same thing. EMA prefers PDEs, how Canada, FDA prefers ADEs. But it's the same thing. It's exactly the same measurement. So just remember that ADEs equal PDEs. What's next? So that means, okay, you evaluated your compound, you did the toxicology assessment on your compound based on literature. Now what's next? So identifying the source of exposure to the different substances, including equipment, processes, and job tasks. This will help to identify where or when workers may be exposed to the substance. Evaluate the current level of exposure to the different substances and compare them to the established HBELs. This will help to determine if the current exposure levels are within safe limits, or you need further control, or further control needed to put in place in order to be at what you already established for the compound. Identify and implement control measures to reduce or eliminate the exposure to the different substances. Control measures may include engineering controls. You have ventilation, administrative controls such as job rotation, and also personal protective equipment, yes, such as respirators and gloves. But again, never base yourself, this is only for accidental exposure, never base yourself on your protective equipment, because often people sometimes misuse their protective equipment. Sometimes they don't change the cartridge properly. Sometimes it's not fitted properly. So you have the last one, the protective equipment, yes, and for accidental exposure, but you cannot only rely on your PPE. Implement the selected control measures and monitor their effectiveness and reducing or eliminating exposure to the hazardous substance. Monitoring may include air sampling, personal monitoring, or biological when you have biological to deal with biological monitoring. Regularly review and revise the control measure as needed to ensure that they remain effective in reducing or eliminating exposure to the hazardous substance. This may include reevaluating your exposure levels, reassessing your control measures. It's a continuous work, right? It's always continuous. You have to make sure that you're always up to date with your things. Like I said, if you evaluate a compound that was evaluated in 2014, we're in 2023. So you need to reevaluate this. You need to reassess. You need to, other indication came out there, new information came out there for toxicology. And make sure that you're still within the limits that were established. Ensure that workers are trained on the proper use of control measures and PPE and communicate any changes or updated to the control measures. Again, of course, if you're doing on your side the toxicological information and you don't communicate this to your workers properly, or they're not trained, some people work very messy. they like to work messy with powders and like, for example, colchicine, don't work messy with this because, you know, it's very potent, 7 milligram, the toxic dose. So you have to make sure, you know, that your personal, because we're doing this to protect them, right? At the end of the day, we're doing this to protect workers. So we need to make sure that on their side, they know what they're doing and they're proper trained. And even for this, for cleaning, the cleaning aspect also of it, it's very important. You have to go see. Sometimes you have to go see in your facility and evaluate your cleaning procedure. Monitor your cleaning procedure. Are they still effective? Are they cutting corners? Or are they really going, you know, you developed a method for a specific compound. So you have to make sure that they're addressing this. And again, as a summary, overall, establishing HBELs in a shared facility requires not only you on your side that does that, it's a responsibility of a coordinating effort that prioritizes worker safety and takes into account the unique challenges of a shared workspace. By following establishing protocols and collaborating effectively, organization can ensure a safe and healthy workplace for all employees. And this is very, I mean, we're trying, you know, we're helping workers, we're trying, and you're helping your business at the same time because you don't want to have recalls and you don't want, you want to avoid all that mess. And for us, it's very dear to heart to protect the workers and to protect the patients at the end of the day, because you're giving this product to a patient that is probably dealing with cancer or probably dealing with, you know, a bad disease that you don't want to make them more sick than they are, right? So again, thank you very much for your time. If you have any question and you want to contact us and you have any other question regarding our products, regarding anything that we can help you, can always contact our cell manager, senior cell manager, which is Peter Burke. We also have Rudaja Dixit, which is the business development manager, and of course our Joanne, which is the business development associate. And again, thank you very much, Joanne, also for all, you know, all your help with this. And it was a very nice, was a pleasure to speak to you guys about this. I think it's a very important, I know it's already a well-known subject, but I think it's good that we go through it again. And if we have to go again and again to sensibilize, you know, the people working in the pharmaceutical industry, I hope I share some light for you guys tonight, and I hope it helped. And it was a pleasure. So again, if you have any question, now's the time.
Yeah, okay. Thank you so much, Christina, for this super insightful session. So I learned a lot also from your session. So yeah, now we'll move on to the Q&A session for this webinar. So you can type your questions down in the chat box and we'll go through them. Yeah, so since there's no questions now, I will start off with my own questions first. So yeah, I wanted to ask about LD50 values. So can I use the LD50 value to determine the health-based exposure limits, or can I use it for like the point of departure?
Yeah, this is a very good question, Joanne. Very good. Honestly, I don't recommend it. I know sometimes we don't have information at all. And in that case, maybe it's your last resort to use. But don't use it. It's not LD50. What is an LD50? It's the median lethal dose. It's based on acute toxicity. It will not give you the long-term effects of the compound. It's not, I understand when you don't have it, yes, you could use it that as the last resource and, you know, increase your factors when you do the calculation because you use, you know, acute toxicity, but it's really not recommended at all.
Okay. Okay. And then another question that I have is that, you know, you mentioned that the European regulatory authorities, they prefer using PDEs, and then like, for example, Health Canada, FDA, they prefer using ADEs. Yeah. So I just wanted to confirm that, like, will all the regulatory authorities accept both ADEs and PDEs, or, you know, do they want any separate things?
It's the same thing. It's exactly the same thing. You know, some regulators are more fancy than others, you know. EMA likes to talk about PD and it's okay. I mean, it's fine. This is the way they do it in Europe. And FDA and Health Canada are talking more often about ADE. But ADE and PDE, acceptable daily exposure and permitted daily exposure, is the same thing. It's really the same thing. You will come to the same numbers. The formulas are a bit like written differently, but the factors put into account are the same. It comes to the if you come to an ADE that, I give you an example, it's 30, and you come to a PDE that is 100, go back and redo your calculations. Something is wrong, because it's the same. It's exactly the same thing.
Okay, thank you so much, Christine. Okay, I see that under the meeting chat, there is no further questions. So, yeah, I mean, oh, there's one question now, sorry. Okay, there's one question. So the question is, I would like to know, does this mean that a PDE report actually comes with an expiration date?
Well, I mean, a PDE report comes to it comes with the reviewing of the compound. Like I said, if your report is done in 2014 and you're in 2023 right now, you need to reevaluate for sure. You need to make sure that you have everything is up to date, and that you don't have any you should review your toxicity information. You should review if other indications were approved out there, if there were other clinical studies done, probably maybe the dosage, because you have a new indication, it's different. So for sure, it needs a review.
Okay. Okay, I have another question. Yeah, so for multi-component drugs, in the absence or the lack of data for establishing the health-based exposure limit as a whole drug, can we use the data for each component instead?
Yeah, like I said, mixtures are very hard to do. Don't forget, we usually do the monographs are based on one active, on the active, because you don't know, is somebody gonna work with one component, or is it come as a mixture already, or someone is gonna wait one compound and will mix the powder, will mix this compound and another compound. So it's always better to do per individual compounds. Because again, don't forget, hazardous equal toxicology property of the compound.
Okay, thank you so much. Okay, another question. So based on your experience, what are the main challenges in establishing health-based exposure limits in a biotech product? And is there any particular difference with chemicals?
Yes. Biotech. Yes, biotech product could be very different than chemicals. Because, for example, I'm just giving you an example, monoclonal antibodies. Or other novel compounds. Some compounds are so big that it will not interact with the cell, so you won't have necessarily required the carcinogenicity study, genotoxicity studies, all that. But again, it's very hard because it's limited. They're novel compounds for an establishing HBELs. So it's a bit different than for chemicals.
Okay, thank you.
The inherent properties of compounds are different.
Right. Okay. Okay. And
sorry, I have a question. Basically, like to chat with you guys. So basically, for example, for a long train, I mean, for train of equipment, right? Does it make sense to have different compound as the most toxic? Does it make sense to have only one compound to be the most toxic compound for example, a bioreactor, or or can it be a case where, for example, bioreactor, the most toxic compound could be this substance A, then the UFSC itself is a compound B when calculating the health-based exposure limit?
Yeah, this I will have to go more like deep in the subject. I cannot really tell you, you should do this or you should do that, because it really, the most most loved expression by toxicologists, and I'm gonna tell you, it depends. It depends.
Okay.
It always depends. It's never black or white. You always have that gray zone, right? But the problem is that, we're doing this right to protect workers. So usually what we're always trying to go with the most restrictive, those descriptors. So for sure, we're going to look at which, what is more, but again, we need to see the literature. We need to see, is it appropriate to go with those descriptors? Sometimes it's not appropriate to go with this, So it depends.
Yep, understand. Okay, thank you.
My pleasure.
Okay, maybe just the last few questions. Okay, one question is, how do we know if the ADE or PDE limits get changed?
How do we know if what do we mean? If AD and PDE get the change, meaning that if it's being reevaluated? Is that what we're saying? If it's being reevaluated?
I think so. Yeah, I think that should be the questions. Yeah, like if it
Okay.
gets evaluated, sorry.
Well, again, we have to evaluate. We have to if you know you're doing a compound and you know recently that this drug was approved for another indication, we need to evaluate. The AD and PD can change. It could be from not potent at all because all the data that were out there was very, was not potent. We didn't see anything, no toxicity, nothing. But now we see that, oh, wow, okay. I'll give you an example, aloe vera. Who thought that aloe vera was carcinogenic? Who thought that? I mean, it's aloe. Everybody's, it's considered safe, right? But yeah, it could change. It could change from time to time. It could change, and it depends on the literature. So you have to make sure that you did a very thorough research, that you know where you're looking at, and that you're experienced enough to know, okay, I have to go look at this, or maybe the therapeutic indication change. I have to go look. You see, maybe the dosage change. So then, if the dosage change and you had a different dose for a different indication, your calculation will based on a different dose, and then your ADE and OEL will change.
Okay. Maybe just one last question. So,
Okay.
Does excipients need to have HBEL?
Why not? Why not? Of course, it's a chemical you're working with, right? For sure, like some excipients are in every tablet or in every drug. Yeah, you have to. It's an excipient. We will evaluate, and some are really low toxicity because they use, some are they have nothing, but you need to evaluate. It's a compound that you're working with in your facility, and you need to evaluate.
Okay.
You need to evaluate because you're going to work with powder. And again, we're doing this to protect the workers. Someone that is working with something unknown, not very safe. You don't know what that person is working with. You need to evaluate.
Okay, right.
If you don't have data, oh yeah, I know this is not fun when we don't have data, but there's always a solution. It depends. We love to say that. It depends. It depends because, okay, you don't have data, but maybe you could do an occupational health categorization of your compound. And that will give you an idea, a range of where your compound should be.
Yeah. Okay. Yeah, thank you so much, Christina, and thank you for all the questions. I'm sorry we can't go through all the questions in the meeting chat, but if there's any further questions, these slides will be shared, and our email addresses and LinkedIn profile will be there, so you can direct the questions to us. Yeah, so once again, thank you everyone for attending this Technical Tuesday session, and thank you, Christina, for sharing your knowledge on this topic. Yeah, so thank you, guys.
Thank you very much. I wish you a wonderful day for someone that it's very early, and a good night for someone that are going to bed.
Thank you.
Thanks a lot. Thanks, guys.
Thank you. Thank you very much, guys.
Bye.
Bye-bye.
Bye.